All right, today we are zooming way in to look at the cell membrane, that living border that literally separates life from non-life. And trust me, it's so much more than just a simple bag. It's a dynamic, intelligent gatekeeper, and we're about to see exactly how it pulls it all off. So let me ask you this. What do you think is the most important gatekeeper in the world? Maybe a bouncer at some exclusive club? A border agent? Well, believe it or not, the answer is microscopic, and there are trillions, literally trillions of them working inside you right now. Yep, it's the cell membrane.
Okay, so the cell membrane's number one job, its whole reason for being, is to create a boundary. It defines what's inside the cell and separates it from everything outside. It is the fundamental line in the sand that makes a cell, well, a cell.
So how do you even build this incredible border? Well, it all starts with this one truly clever molecule called a phospholipid. It's what scientists call amphiphilic, which is just a fancy way of saying it has a total split personality when it comes to water.
And this slide just lays out that split personality perfectly. On one side, you have the happy, water-loving head. That's the hydrophilic part. It can't get enough of water. And then you have these two tails that are absolutely terrified of water. That's the hydrophobic part. They will do pretty much anything to get away from it. And this is where the magic happens. This process of self-assembly is just incredible. The moment you drop these phospholipids into water, those water-hating tails desperately scramble to hide from it, clustering together on the inside. At the same time, the water-loving heads happily face outward, towards the water. And just like that, you get this stable, two-layered sheet, the phospholipid bilayer. It builds itself. But here's the thing. The membrane isn't just a wall. It's more like a super-selective bouncer at the world's most exclusive club. This is a concept called selective permeability. It has a very strict guest list and gets to decide exactly what comes in and what gets kicked out.
So who actually gets past this bouncer? Well, as you can see here, it's all about two things: size and charge. Little uncharged things like oxygen, they can just stroll right on in. Water is small, but it has a slight charge, so it can kind of sneak by, but slowly. But then look at something like glucose. It's big and it's polar. It's pretty much stuck outside. And charged ions like sodium? Nope, the door is slammed shut. Absolutely no chance of getting through this lipid layer on their own.
So wait a minute. If super important things like glucose and ions can't get through, how on earth does a cell even survive? Well, that's where the real specialists come in, the proteins. You see, while the phospholipids give the membrane its basic structure, it's the proteins embedded inside that do most of the active, really crucial jobs. Now, these protein workers come in three main flavors, all based on where they hang out. You've got the big guys, the integral proteins, that go all the way through the membrane. Then you have peripheral proteins, which just chill on one side or the other. And some are even tucked right into the bilayer itself.
Okay, for getting stuff across the border, the most important workers are the transport proteins. And they come in two main types: channels and carriers. Channel proteins are like simple tunnels. They just open up and let specific things like ions rush through. And when these go wrong, it's a huge deal. It can lead to diseases like cystic fibrosis. Carriers, on the other hand, are way more sophisticated. This shows you exactly how a carrier works, using the GLUT4 transporter as our example. Check it out. In step 1, the outer gate is open, just waiting. Then, a glucose molecule comes along and fits perfectly into its binding site. This triggers a change. The outer gate slams shut, and the inner gate opens up, releasing the glucose inside the cell. It's this perfect one-by-one delivery service.
Then you've got the real power lifters of the membrane, the pumps. These proteins use the cell's energy currency, a molecule called ATP, to do some serious heavy lifting. They actively pump things against their natural flow, from an area of low concentration to high concentration. Think of it like trying to push a giant boulder uphill. It takes a ton of energy, and the famous sodium-potassium pump is the perfect example.
Proteins are also amazing structural anchors. These cell adhesion molecules, or CAMs, their whole job is to reach out and hold on to the cell next door. You can think of them like biological Velcro, sticking cells together to form tissues. Some of them form even tighter connections, acting like waterproof rivets that create a perfect seal. It's absolutely essential for keeping our organs together.
So beyond just moving and anchoring, proteins also have to listen. Cell surface receptors are basically the cell's mailboxes. They sit on the surface just waiting for a specific chemical signal, like a hormone, to arrive. When that letter shows up and binds, it triggers a message on the inside. And by the way, this is exactly how a lot of nasty viruses, from the flu to HIV, trick their way into our cells by hijacking these very same mailboxes.
And I absolutely love this description. When your pancreas releases insulin because you just ate something sugary, that insulin is a message. It binds to a receptor on a cell, and the whole process is like the cell getting a love letter from the pancreas that says, open the door to get a sweet treat. How cool is that? This signal tells the cell to bring more of those GLUT4 transporters to the surface to start bringing in all that sugar. It's communication on a molecular level.
Okay, so we've got the phospholipid structure and the protein workers. But there's a supporting cast of other molecules that are totally essential for the membrane to work the way it's supposed to.
Let's talk about cholesterol. This molecule is a genius because it plays this amazing dual role. Think of it as a temperature buffer. When things get too cold, it squeezes in between the phospholipids to keep them from packing too tightly so the membrane doesn't get brittle and crack. But when it gets too hot, it does the exact opposite. It pulls the phospholipids together to keep the membrane from becoming too leaky. So, cholesterol makes the membrane both fluid and durable.
And the final touch is carbohydrates. You'll find these attached to proteins and lipids on the outer surface of the cell. Think of them as the cell's unique ID cards or name tags. They're what allows cells to recognize each other, and really importantly, they're how your immune system can tell the difference between your own healthy cells and a foreign invader.
So we've met all the individual players, right? We have the lipids, the proteins, the cholesterol, the carbs. Now let's put them all together to understand the complete picture, which scientists describe as the fluid mosaic model.
So here's our cast of characters one last time. The phospholipid bilayer is like a vast fluid sea. The proteins? They're like giant icebergs of all different shapes and sizes, just floating around in that sea. Cholesterol is the stabilizer, making sure the sea doesn't freeze or get too runny. And the carbohydrates are like little antennas sticking up from the icebergs, broadcasting the cell's identity to the world.
And this brings us right to the central idea. The membrane is not a static, rigid wall. The name fluid mosaic model tells you everything you need to know. It's a mosaic because it's built from all these different pieces. And it's fluid because those pieces are constantly drifting and moving around each other, kind of like boats on a lake. It's a living, dynamic, ever-changing surface. And just to hammer home how important those protein icebergs are, get a load of this. By mass, proteins make up about half of the entire cell membrane. 50%. That's insane. It just tells you that what the membrane does, all that transporting, signaling, and anchoring, is just as important as what it is.
So, if you take one thing away today, let it be this. The cell membrane is not a simple container. It's a bustling, intelligent, flexible border. It's a living interface that is constantly sensing, communicating, and working non-stop to maintain that delicate balance we call life.
Which really just leaves us with one final, kind of mind-blowing thought, doesn't it? If this one microscopic border, this tiny gatekeeper, is so incredibly complex and active, just imagine what other incredible machinery is humming away right now inside every single one of your cells.